Hello friends, welcome to bioassembling. In this session, I will discuss about ribosome and their structure and function. Let's start the session. First of all, we understand ribosome, their structure and function. Ribosome first discovered by G.E. Palade in 1952. Means ribosome are protein synthesis unit of cell and described by G.E. Palade in 1952. Ribosome are complex of ribosomal RNA and various protein. Means ribosome contain of ribosomal RNA or rRNA and various type of proteins. Ribosome are small, dense, rounded and granular particles of the ribonucleoprotein. Means ribosome are small and rounded granular particles which contain ribonucleoprotein. Next, we understand where ribosomes are present. First of all, they occur either freely in the matrix of mitochondria, chloroplast, and cytoplasm, or remain attached to membrane of the endoplasmic reticulum. When ribosomes are attached to endoplasmic reticulum, they are called rough endoplasmic reticulum. They occur in most prokaryotes and eukaryotic cell and provide scaffold form of order of interaction of all the new molecules involved in protein synthesis. Means ribosomes form scaffold orders and interact all ribosomal molecules and form protein synthesis. They are most abundant RNA proteins complex in the cell which direct elongation of polypeptide at the rate of 3 to 5 amino acid added per second. Means complex when the protein are synthesis when the amino acid attached to form polypeptide chain and where the rate of polypeptide attachment is 3 to 5 amino acid per second. Small proteins of 100 to 200 amino acids are therefore made up in a minute or less. When small protein are formed, it takes 1 minute or less time. It takes 2 to 3 hours to make the largest known protein, titin, which is found in muscle and contains about 3000 amino acid residue. Largest protein making process are to take 2 to 3 hours, and these are found in muscles and it contains 30,000 amino acid residue. Next, when we talk about the types of ribosome are mainly two types, 70S ribosome or 80S ribosome. Ribosome are classified into two types based on their sedimentation coefficient. 70S and 80S ribosome classified to two types and on the basis of their sedimentation coefficient. As Stanford Sedberg unit and related to sedimentation rate, means it depends upon the sedimentation mass and size. Thus, the value of before S indicates size of ribosome. Next, 70S ribosome. Prokaryotes have 70S ribosome. The 70S ribosome are comparatively smaller in size and have sedimentation coefficient is 70S with molecular weight is 2.7 into 10 to the power 6 daltons and means prokaryote have 70S ribosome and 70S ribosome are comparatively smaller in size with molecular weight is 2.7 into 10 to the into 106 daltons. When we see a ribosome with electron microscopy, the measure of dimension of the 70S ribosome is 170 into 170 into 200 AO, and they occur in prokaryotic cells of the blue-green algae, bacteria, and also in mitochondria, chloroplast of eukaryotic cell. This all about the 70S ribosome. Next we discuss about 80S ribosome. 80S ribosome. Eukaryotes have 80S ribosome. I will repeat, that means prokaryotes contain 70S ribosome but eukaryote contain 80S ribosome. 80S ribosome have sedimentation coefficient of 80S and molecular weight is 40 into 10 to the 40 into 106 daltons. And the 80s ribosome occur in eukaryotic cell of plants and animals. The ribosome of mitochondria and chloroplast are always smaller than 80s cytoplasmic ribosomes and are comparable to prokaryotic ribosome. That means ribosome of mitochondria and chloroplast is smaller than 80s cytoplasmic ribosome and are comparable to prokaryotic ribosome. Prokaryotic ribosome in both size and sensitivity to antibiotics. However, their sedimentation value vary in different phyla. 77S in mitochondria in fungi, 60S in mitochondria of mammals, and 60S in mitochondria of animals. This all about the 80S ribosome. When we talk about the chemical composition of ribosome, the ribosomes are chemically composed of RNA, proteins, their major constitution both occurring in approximately in equal proportion in smaller as well as in larger subunit. 70S ribosome contain more RNA 60 to 40% than the proteins 36 to 37%. Means ribosome chemically composed of RNA, proteins, and its major constituents are RNA and proteins, and equal proportion of smaller as well as larger subunit. 70S ribosome contains 60 to 40% RNA and protein is 36 to 37%. The ribosome of E. coli contains 63% rRNA, 37% protein. Means E. coli contains 63% rRNA and 37% protein. While the 80S ribosome contain less RNA, 40 to 44% than the protein 60 to 56%. Yeast ribosome has 40 to 44% RNA and 60 to 56% proteins. Ribosome of pea seedling contain 40% RNA and 60% protein. There is no lipid contained in ribosome, means ribosome do not contain any lipid. Let's talk about ribosomal RNA or rRNA. RNA constitute about 60% of mass of ribosome. The 70S ribosome contains three types of rRNA. Means 70S ribosome contains three types of rRNA: 23 rRNA, 16S rRNA, 5S rRNA. The 23 and 5S rRNA occur in the larger subunit 50S of ribosomal subunit. Means ribosome are contain two subunit 50S and 30S in 70S ribosomal subunit. While 16S rRNA occur in smaller 30S ribosomal subunit, the 80S ribosomes contain four types of rRNA. rRNA in 80S ribosome are four types: 28S rRNA or 25 to 26 rRNA in plant, fungi and protozoa. 18S rRNA, 5S rRNA, 5.8S rRNA. The 28S, 5S and 5.8S rRNA occur in larger subunit or 60S ribosomal subunit. Means 80S ribosome contain two subunits 60S and smaller subunit are 40S. Means 28S, 5S and 5.8S rRNA occur in larger subunit or 60S ribosomal unit, while the 18S rRNA occur in smaller subunit or 40S ribosomal subunit. About 60% of rRNA is helical, means it forms double helix structure like DNA structure and it paired with bases. These double stranded region are due to the hairpin loops. It forms hairpin loops between complementary region of the linear molecules. This all about the ribosomal RNAs. Next I will discuss about the structure of RNAs molecule. RNA molecule contain larger subunit and the smaller subunit. This is larger subunit and this is smaller subunit, and it contain codons. Three codon 1, 2 and 3. And this is tRNA or transfer RNA. This is exit site And these are mRNAs and terminus, means when uh, protein molecules are formed, then it terminates the formation of proteins molecule. And this is mRNA, 3 mRNA terminus, this is 5 mRNA terminus. These are the mechanism of protein synthesis. Here, first of all, amino acid residues are attached to tRNA or transfer RNA in codon 1, and this process is called elongation. And when repeated type of amino acid is attached to codon 2, and it form polypeptide growing chain, and this messenger RNA are moving and going to attach the amino acid, and new transfer RNA is attached to codon 3, and this process is repeated and the polypeptide chain form, and the process of when new protein molecule is released, is the process is called termination. These are the polypeptide binding site, you can see this structure, and these are exit site. When the protein molecule are formed, then this is called exit site. With the help of exit site, it's released from the ribosome. And these are amino acyl binding sites. Later, this all about the structure of rRNA. Next, I will discuss in upcoming lecture about the process of protein synthesis or translation. So this all about the ribosomes and their structure. When we talk about conclusion, first of all we study about the who discovered ribosome, GE Paladin 1952, and where ribosome occur, mitochondria, chloroplast, and endoplasmic reticulum, and prokaryotic and eukaryotic cells. Next, I will discuss about the type of structure, and which is 70S ribosome, and which occur in prokaryotic cells. 70S ribosome are comparatively smaller, which molecular weight is 2.7 into 106 daltons. Next, 80S ribosome. 80S ribosome occur in eukaryotic cell, with molecular weight is 14 to 10 to the power 6 daltons. Next, I will explain about the chemical composition of RNA, which is protein, RNA, and major constitutes. Next, I will also explain about ribosomal RNA, which is 60% of mass of ribosomes, and ribosomal RNA or rRNA, which is in 70S, 23S, 16S, and 5S. And in 80S ribosome contain 4 type of rRNA, 28S rRNA, in plant and fungi 18S rRNA, 5S rRNA, and 5.8S rRNA. These all are occur in 60S ribosomal subunit, and 18S rRNA occur in 40S ribosomal subunit. And next, I will discuss about the structure of ribosomes, which contain larger subunit and smaller subunit, larger subunit and smaller subunit attached in form 3 codons, codon 1, 2, and 3, and these are exit site. These are where the polypeptide chain are grow and attached to form protein molecule. When the protein molecule form this exit site, the protein molecule release from this exit site, and these are the binding polypeptide binding sites, and these are the growing polypeptide chain. This all about the structure of ribosome. So this all about ribosome. Thank you so much for your watching. Please like, share, and subscribe.